know that even today, measles results in 100,000 deaths per year across the globe, and that measles is associated with increased morbidity and mortality for years after the initial infection, but we've never totally understood why. It turns out the measles virus receptor is highly expressed on memory, TB, and plasma cells, leading to their depletion. And although these cells appear to recover in numbers, it's been hypothesized that measles virus actually induces what's called immune amnesia, or simply impaired immune memory. So this study in Science tried to address this question. And what they did is they used a really novel method called VIR-scan. And this particular method tracks the antibodies to thousands of pathogen epitopes in the blood. And these pathogen-specific antibodies detected in the blood are thought to represent changes in these long-lived memory repertoire responses of the immune cells. So they studied several populations, so it was a little complicated, but the key population was a group of children that were unimmunized and who developed measles in an outbreak that was in the Netherlands. And then they included a number of control groups that were children that were evaluated at 2 different time points for comparison, as well as some adults that were evaluated, some unimmunized and uninfected children that were within the same outbreak, and 33 children who had received the vaccine. So on the slide at the top are shown the key results. And on the left of the top graph are the 3 control groups looking at these antibody responses at 2 different time points. And you can see in the controls, these epitope-specific antibody responses are pretty similar at 2 time points. This was also true in the next bar, where the measles virus negative group. But if children got the measles vaccine, you see an increase in the epitope recognition, which contrasts markedly with the antibody levels in children who either had mild or severe measles. And overall, they saw a 20% decrease in the diversity or size of this antibody repertoire. And this was without seeing any changes in the overall immunoglobulin levels. When they actually focused on epitopes that they evaluated specifically in the pre and post sera, the declines in this repertoire were even more severe. In the heat map on the bottom of the slide is another display of these results. And now on the left, you can see just a smattering of the antigens that they looked at. And when you look at this, you can see in the control group that there's a mix of blue and red, with red being stronger responses and blue being weaker responses to these antigens. In the children who didn't have measles virus, you can see there's no antibody response at the top to measles, but otherwise those children look like controls. Similarly, the children who got the vaccine, you can tell because they have a strong antibody response at the top of the graph to measles and rubella, but then the rest of the coloring looks very similar to the controls. And you can contrast that to the last 2 groups, who are children who had measles, who have a very strong red response to the measles virus, but a blue response predominantly to these other antigens, which displays that they had less antibody responses. So it's not only the number of epitopes recognized, but the strength of the epitope response that is diminished after measles infection. And importantly, from this graph, the measles vaccine did not impair these responses. So overall, these findings underscore the importance, which all of us know, of the need for continued widespread measles vaccination. And I'll just remind you that in 2019, WHO estimated that measles virus vaccine prevented more than 21 million deaths between the years of 2000 and 2017. Thank you very much for your attention.